In this video, I'm going to continue our discussion on calibration by looking at calibration methods, and at the end, I'll talk a little bit about calibration curves in general. Now, for a lot of this video, I'm going to be using the worksheet, but I will switch back and forth in between some of the slides. So the first calibration method I want to talk about is one that you are the most familiar with and you've used both in lab and probably in other chemistry courses. It's called the external standards calibration method. So basically in this method, what you would do is to prepare a stock solution, for example, a 100 ppm iron solution, as I've shown here. And from that solution, from the stock solution, you would take specific volumes, say 1, 2, 3, 4, and 5 milliliters, as shown in each of these volumetric flasks in red, and then you would fill the volumetric flask up to the mark with whatever solvent that you are using. Usually in analytical chemistry, that would be water. So the prepared standards are completely different from the unknown. They're completely separate because the unknown itself on the right contains only the unknown. Now the unknown may need to be diluted, such as in the iron vitamin experiment, right? You were given 10 milliliters of the unknown and then you had to dilute it to 100 milliliters. But the unknown itself was completely separate from the standards. And you can see down at the bottom here, the volume of the unknown. There is nothing in the standards, and there is a certain amount, 5 milliliters in this case, in the unknown. So this is one of the differences that you will see between this method and the next method when we get there. So using this method, the calibration standards are usually clean, meaning that the matrix is not very complex. It's quite simple. For example, it might just be water. It might be water and a few other reagents that are required for a color change or some other purpose. And the unknown sample contains the analyte, reagent, and the matrix. So remember in the last video I was talking about how important it is to match the matrix of the sample to the matrix of the standards. In this case, that would be difficult to do because we would have to prepare in the lab something that would match the matrix of our sample. So this method is usually only used for samples that have a very simple matrix. And to determine the concentration of an analyte using this particular method, you would first prepare a calibration curve as shown here on the right. And then if you had a specific signal of unknown that was, say, right here, you would use the equation of the line to figure out what the concentration of the analyte was. So this is the equation that is given in your book. It looks like this, but more commonly you might see it like this. It's the same equation, just in your textbook it gives it slightly different variables as it tries to explain what each of those variables actually is. So the next method I want to talk about is the standard addition calibration method. And this is the method to use when you think that something in the sample is going to affect or interfere with the analysis. And usually this would happen when the matrix is complex. So when you're preparing standards using this method, the difference is that all of the standards contain the unknown. So the unknown here shown in orange. And they all contain the same amount of the unknown. In this case, each of those flasks contains 5 milliliters of the unknown sample. And then the standard addition part is where you take your prepared standard, the 100 part per million iron standard, for example, and then add that in different amounts to all of the samples. So first sample doesn't have any iron standard in it, so it is only the sample itself. The next standard has the sample plus a small amount of the 100 ppm iron standard, and the next sample has a little bit more of the iron standard, and so on and so forth. So each of these samples contains the unknown, and all but one of these samples contains the iron standard. So it's quite a bit different than the external standard calibration method, and the reason that we do this is so that we can match the matrix of the sample with the matrix of our standards, because we're putting the sample right inside our standards, so the matrix is automatically going to match. And this is going to be important if we have a complex matrix, because anything that's going to interfere with the signal is already going to be present in all of our standards. Now I want you to take a look at what a calibration curve looks like using the standard addition calibration method. Notice that the first point is not at 0, and the straight line also doesn't go through the origin, right? The origin being right here. Because the sample that we analyze first, the one that doesn't have any iron standard in it, still has the sample, and the sample is going to have the analyte. So the analyte itself is going to give us some kind of a signal. And then by adding different amounts of iron standard to each of the other flasks, we're going to get increasing signal from those standards. And the way to deal with this type of a calibration curve when we're trying to figure out the concentration of the unknown is to first extrapolate the line backwards to a point where y equals 0, and we're going to solve the equation of the best fit line for x. So we're going to set y equals 0 and solve for x. And the other difference in this calibration curve is that on the x-axis, we don't have a concentration. Well, not in this case. It's possible that we could put a concentration down there, but usually we're putting a volume of standard added. So in this case, if we look back up at the dilution scheme, the first added standard is 2 milliliters, the second is 4, the third is 6, and so on and so forth. So down at the bottom of this calibration curve, we would have the volumes of added standard. So using the equation of the line, we set y or S spike to 0, so the signal of the spike to 0, and solve for x, or the volume of the standard. And then this gives us a number. It's a negative number, but that volume, if we make it a positive number, would be the volume of the standard that would be required to give us the same signal as the unknown sample. So if we were to take a sample that had only the matrix, it didn't have any analyte in it, and we were to add that specific volume of our standard into that matrix, then we would get the same signal that the unknown itself gives us. And we can use that information to determine the concentration of an unknown sample. So we do that by multiplying the concentration of the standard, which is usually given. We know what we're making, and we know how we're making those standards, right? We know how we make the 100 ppm iron standard. So we can multiply that standard concentration by the volume of the standard that we got from the calibration curve. So the volume that we got from solving the equation of the straight line for x, right? So y equals 0, we'll solve for x. And then we divide that by the volume of the unknown. So depending on how much unknown was added, in this case we added 5 milliliters of the unknown. So we would divide the concentration of the standard times the volume we get from the calibration curve, divide that by the volume of the unknown, and that's going to give us the concentration of our standard. Now, if you think about this, this is really a CO-VO equals CD-VD calculation, where CO is the concentration of the standard, VO is the volume that we get from the calibration curve, VD is the volume of the unknown that was put into our sample, and CD is what we're going to be solving for. So I'm going to go through an example of a standard addition calculation so you know how to solve that type of question. So here's the question from the slides, and the graph that I show here is also on your slides as well. So the question refers to this figure, and it gives you an idea of how each of the solutions were prepared. So we're trying to determine the concentration of manganese in an unknown sample, and each solution contains 25 milliliters of the original sample and either 1, 2, 3, 4, or 5 milliliters of a 100.6 milligram per liter external standard of manganese. So all standard addition samples were diluted to 50 milliliters before reading the absorbance, and the equation of the line is given, and we're asked to determine the concentration of manganese in the unknown sample. So the first step is to determine what point is here. We're going to determine the volume of standard that would be required to give us the signal for the unknown. So in that case, we're going to set y to 0 and solve for x. So if we write that out, we're going to have 0 equals 0.0854x plus 0.1478. And solving for x, 0 minus 0.1478 over 0.0854 equals x, and then x is going to equal minus 1.7307 milliliters. Now your book handles this a little bit differently than I'm going to handle it, but basically what I like to do is, instead of keeping this volume negative, I'm just going to make it a positive number. So we're going to call it 1.7307 milliliters. And again, that is the volume of standard that will be required to give that signal. Okay, remember that signal that I'm pointing to is the signal for the unknown sample itself. But if we could remove all the manganese from the unknown and simply add this volume of standard to that particular matrix, then we would get that same signal. Okay, so now in order to solve the concentration, we're going to take the volume that we just solved for, 1.7307 milliliters, and then we're going to multiply that by the concentration of the standard, which was 100.6 milligrams per liter. And what this is going to give us is the milligrams of manganese in the unknown. Now if we continue our COVO equals CDVD calculation, on the right hand side of the equation we're going to want to solve for the concentration of the sample. And the volume of the sample in this case was 25 milliliters. Right? Because in the question it said that 25 milliliters of the manganese unknown was used, and that volume was diluted to 50. But we don't care about what the concentration was in the 50 milliliter solution, because that's our dilution. We want to know how much is in the 25 milliliter solution, which would be the original sample. So the concentration of manganese in the original unknown sample. So again, this is just the volume of the unknown. And if we solve for the concentration of the sample, that's going to equal 6.96 milligrams per liter. So that would be our final answer. So that's the concentration of manganese in the unknown solution. So the next method I want to talk about is called the internal standards calibration method. Now it's very common that students will mix this method up with the standard addition method. So try and separate these in your mind because they are quite different. So the standard addition method, the method that I talked about previously, is used when we have a complex matrix and we want to make sure that we can match the sample matrix with the standards. For internal standard calibration method, we use this when we want to control for variations in the signal due to factors that are difficult to control, such as electrical fluctuations in our instrumentation, or when we're doing sample preparation such as digestion, filtration, or extraction, where sample loss is likely. Another reason to use the internal standard calibration method is if we are doing an analysis that requires us to inject some sample into our instrumentation, and it's difficult to inject the same amount of sample each time. So even if we used the same sample and injected that sample 3 times, we might get different signals for that particular sample. And if we didn't know it was the same sample, we might think that it's 3 different samples that have 3 different concentrations. But the internal standard can help us determine that it is indeed the same sample, and we can use the internal standard to correct for any fluctuations in the signal. And I'll explain that in just a second. So in the example I'm going to use here, we have ethanol as our analyte, so that's what we're trying to measure. We have propanol as the internal standard, and we have an unknown, which is going to be an ethanol solution. So notice in this particular example, it's quite similar in some ways to the external calibration method. So if you ignore the purple for now, that's our internal standard, but just ignore that for now and just look at the blue. So the blue kind of looks, well, almost exactly looks like how we would prepare the external calibration standards. We have nothing in the first sample, so that would be the blank, and we have increasing amounts of ethanol that we would put into each of the standards. So in this case, we have 0, 2, 4, 6, and 8 milliliters of the ethanol. So the ethanol amount is changing because that is the analyte that we are looking for, and we need to know how the signal changes when we have different amounts of ethanol in solution in order to figure out what an unknown concentration would be. Now focus on the purple, which is the internal standard. And notice that the same amount of internal standard is put into all of the flasks, even the unknown sample. Right? So ethanol changes because that's our analyte. Internal standard stays the same. It doesn't change. The volume is the same in all of the samples, but it is also present in all of the samples. So in a way, I guess this is similar to how the standard addition works, because in the standard addition method, we have the same amount of unknown in each of our standards, but in this case, it's not the unknown that's in all of the samples, it's the internal standard. The unknown is still completely separate, just like the external standard method. It's completely separate from the standards. So the volume of the unknown, 0 in all of the standards, again because they're separate from the unknown, and then a certain amount of unknown is used for the sample itself. So the calibration curve for this looks very similar to the external standard calibration method, but the one thing that is different is what is on the y-axis. So the y-axis for the external standard calibration method is simply the signal of the analyte. But in this case, for the internal standard calibration method, we are measuring the signal of both the analyte and the internal standard. And the y-axis is going to be a ratio of those 2 signals. So essentially what this is doing is it is using the internal standard to correct for any differences in the signal from the analyte. The internal standard is going to be a compound that has very similar properties to the analyte itself, because we want the internal standard to act in a similar way as the analyte does when we're going through our method. So in the case of our example, we have ethanol as the analyte and propanol as the internal standard. They're both alcohols, they both have high vapor pressures, they're both similar in size, they have very similar properties. So how does the internal standard actually help us correct for any differences in the sample? So I'm going to draw a couple of graphs here which will hopefully help this make sense. So if we were doing an analysis of ethanol and we use propanol as the internal standard, let's just say that we're measuring the same sample. We're going to get a peak for ethanol and then another peak for propanol. Now, if we ran the same sample again, you might expect everything to look the same. So I'm just going to we'll call that a signal of 1 and this a signal of 2, just to make it simple. So the signal of 1 and 2, so the signal of 1 and 2 being at the very peak of each of those curves. Now, if we did the same analysis, maybe we get something that looks a little bit like this. So we have another peak of ethanol, but it's a bit bigger than the previous analysis. And then propanol, that peak is also bigger than the previous analysis. Now we might think that these are two different samples because it looks like there's more ethanol in this particular sample than there was in the previous one. But because we have the internal standard, we can rule that out. So let's think about the sample itself. It's going to have a specific concentration of ethanol, and that concentration is not going to change. It's going to have a specific concentration of propanol as the internal standard, and that concentration isn't going to change. We're not doing anything to the sample. All we're doing is an analysis of the sample. So if you think about the concentration of ethanol being constant, the concentration of propanol being constant, hopefully it's obvious that the ratio between those two concentrations is also going to be constant. It's not going to change. So although these two analyses seemingly gave different concentrations of ethanol and propanol, because the peaks were different heights or different peak areas, because the ethanol increased, the propanol also increased to the same amount, and the ratio between those two peak areas is going to remain constant. So that's why the y-axis over here is the ratio of the signals, because the ratio is not going to change, even if the signals itself changes. So just a quick review of the three different methods we talked about. The external standard calibration method, where we have separate standards that are prepared from a stock solution, and each of those standards is made separately from the unknown. Then we have the standard addition calibration method, where each of the samples contains the same amount of the unknown and different amounts of the standard. And then the internal standard calibration method, where the samples and the standards are again separate, and each of the samples contains the same amount of the internal standard. Even the unknown contains that amount of internal standard, and the standards themselves contain different amounts of the analyte. So I want to quickly talk a little bit about calibration curves and the different parts of a calibration curve and what's sort of important about them for our specific uses as analytical chemists. So on the figure to the right shows what a calibration curve might look like if we did an analysis from very low concentrations to very high concentrations. Now, most of the calibrations you've seen, if not all of them, have only been straight lines. But if we start low enough and go high enough in concentration, eventually the calibration curve will deviate from linearity. And that's because there's limitations on how each of the methods are able to detect specific concentrations. They're very linear over specific concentration ranges, but above that range and below that range, they're no longer able to have a linear response. So we've already talked about limit of detection and limit of quantitation. Again, limit of detection is the smallest amount of analyte that a method can see or detect. The limit of quantitation is the smallest amount of analyte that a method can reliably quantitate. We have a linear range, which I think is pretty self-explanatory, and that particular range is usually where we want to work because the change in signal is predictable as the concentration is changed. The limit of linearity, the LOL, is the part of the calibration curve after which the calibration curve is no longer linear, and the dynamic range is the range over which we can reliably quantitate the analyte. All right, so again, we are very much interested in the linear portion of the curve because that's where concentrations and signals have a predictable relationship. So after we do an analysis and get a calibration curve, we may end up with calibration curves that look like any one of these three calibration curves. We have good, bad, and ugly. So a good calibration curve is where all the signals from the standards lie on a straight line. Now there probably will be some variation, but that is still pretty much a straight line here on the left. Now the second one is not all bad. What's bad about it is how the straight line is drawn through that. When you're drawing a straight line through a set of data points, remember that you should only use the linear portion of the curve. You can't draw a straight line through a curve, so don't even try to do it. So while this is bad in terms of how the straight line was drawn through it, it's not necessarily a bad calibration curve, because you might be able to draw a straight line through, say, 4 of those points. And those 4 points would make up the linear range, and that's how you would want to draw a straight line through data that curves like this. So you want to exclude any points that deviate from linearity. So in this case, I've chosen these 4 points as deviating. Maybe only 3 points actually deviate, but we'll say in this case that we have 4 points that deviate. Don't include those in your straight line determination. Use only the first 4 points in this case, and that ends up being a good calibration curve because we're only using those 4 points. Now, the ugly one, there is no way that we can draw a straight line through that data. So that type of data just isn't useful at all. So that's all I have for calibration. Hopefully you can distinguish the difference between external standard calibration method, standard addition calibration method, and internal standard calibration method. And I also hope you understand what a good calibration curve looks like, the different parts of a calibration curve, and how to draw a best fit line through a set of data points. And as always, if you have any questions, please feel free to ask.